Good afternoon. Thank you for coming today. I'm Akihisa Yamamoto, working here as a postdoc in iCeMS. And today I'm going to talk about something sticky in your body and something sticky in biology. And to start that, I want to show you this toy. It's Lego. When I was a kid, my mother bought it to me. And today I brought it from my friend. So what you can do with this is basically you can build up whatever you want by using these tiny blocks. And there are some worldwide artists who are called Lego masters, and they can make something like this. It's really complicated, but if you're good at it, you can make it. And I was so into it, I was playing all the day with these Lego blocks. But before that, before getting these Lego blocks, I was playing with these wooden blocks. Of course, you can also make something nice. You can construct something, but there is a big difference between these two. So when I made up something with Lego blocks, then my parents told me, this is pretty cool, so why don't you put it somewhere? So I could bring it to somewhere at a safe place, and I could, exhibit my art. So it was pretty nice. But on the other hand, it's not the case with these wooden blocks, because you can't bring them. It's not stable if you want to bring them. So what's the difference? So difference is coming from these bumps, as you can see here. So these bumps and also holes at the bottom is making it possible to connect these blocks, which is not equipped in the wooden blocks. So the big difference here is that you can connect these blocks, and this connection helps you to have the stable structure. And you have also something similar in your body. So if you look at inside your body, you have several kind of different organs. And these organs, if you take a look closer, then you have these kind of patterns. And these patterns are all coming from cells. So cells are spontaneously putting together, and they form this kind of patterns. And these patterns are so-called tissues. So tissues are functioning to express the organ functionalities. And each organ has the different function, and also the pattern is specific and unique for each tissue. So this is quite important feature, and in this sense, cell is kind of similar to Lego blocks. But sometimes, even in the same tissue, you may have the different patterns. For instance, when you get ill. So this is the example. So this is the tissues taken from human stomach. And one is from healthy tissue, and the other one is from metastatic tissue. Metastasis is the later stage of the cancer, so it's actually quite severe. And now I'm going to ask you, which of image do you think is metastatic and which is healthy? Can you guess? So if you think the left image is metastasis, please raise your hand. Left, it's metastatic. Thank you. And if you feel the right one is the metastasis, please. Thank you very much. So here is the answer. So the right one is the metastasis. So when you get unhealthy, sometimes the pattern is lost. And this test, what we have done now, is exactly the same as what the medical doctors are doing in the hospital on the daily basis. So they know how the healthy tissue should look like, and if it's gone, then from this changed pattern, they can judge how severe the cancerous stage is. So it's known that the pattern is somewhat related to your health. And if you lose the pattern, you lose your health. But why? We don't know the reason of it. And that's what we are going to focus as a research. And the one keyword is their stickiness. So what happens if stickiness is changing? This is our research topic. So now coming back to the tissue image. So the tissues, they are composed of cells. And if you think about the contact between cell and cell, it's like this. So the cell surface is looking like this. There are lots of molecules inside, and the cell surface looks like the membrane. And when the cells are touching each other, these lots of molecules are interacting with each other. And since there are lots of different molecules, there are lots of different interactions, such as adhesion molecules and polymer interaction and electrostatic interaction by charges and the interaction coming from the fluctuation of the membranes. So all of these chemical and physical originated interaction gives you the force. They generate the force. And the important thing here is that the balance, the interplay of these forces determines the adhesion of cells. If the force is well balanced, then your cells will be kept at a certain distance, so that cells will be kept as a contact state. So it's quite important to understand where the adhesion is occurring and how strong it is. To characterize this adhesion, we are using the techniques based on visualization of adhesion and the direct measurement of adhesion. So today I'm going to introduce these two techniques, how we can do the visualizing and the measuring the adhesion itself. And I want to emphasize that to deal with cell-cell contact directly is still quite a tricky thing. Suppose that you have different kind of cells which has different adhesion property on the top of these images. If you want to know the difference of the adhesion property, basically you would expect to have the cells on the bottom which has the identical adhesion property, because adhesion is mutual thing. So if the adhesion property of the bottom cells is different to each other, you can't distinguish what is the contribution of the top cells to change the adhesion. So you need some kind of standard stickiness to characterize the adhesion that you want to focus on. So for this purpose, we use, instead of using real cell-cell contact, we use so-called artificial cell membrane. This is something that you can really easily prepare in the lab. And the advantage of this artificial cell membrane is that you can tune the molecular composition as you want, and you can use the adhesion molecules as you want. So the stickiness of this artificial cell membrane is fully controllable. So by putting cells on it, it's quite similar to real cell-cell contact, but you have the control on the one side in terms of adhesion. This makes us possible to investigate the characterization of adhesion of cells by having standardized stickiness. So throughout all the experiments, we utilize this technique using artificial cell membrane. And now I'm going to introduce how to visualize the adhesion. So for this purpose, we utilize the physical phenomenon so-called interference. Interference is the overlapping of two or multiple waves from different wave sources. So sometimes by looking at the different position, you have the higher amplitude of wave. Sometimes you have lower amplitude wave compared to the original wave. And we utilize this technique, this phenomena with the light, because the light is also the wave. So if you observe the cells with the light, this is what happens. So we shine the cells from the bottom, and at each surface, at the glass substrate and the cell surface, this light will be reflected. And all of these reflected light makes the interference. And since this is the light, interference results in the change in contrast. So sometimes you have the brighter light, sometimes you have the darker light. It depends on the height of cell surface from the substrate. So this is only parameter to change the contrast or brightness of the interfered light. And we use this technique equipped with the microscope. Therefore, this technique is so-called reflection interference contrast microscopy, RICM. So this is how to visualize the adhesion. And when you look at cells with the conventional method, this is again healthy cell and metastatic cell from the human stomach. They look quite similar. They are just both round. But by observing these with RICM, it's pretty much different. So from the healthy cell, you get this kind of dark patches, while metastasis cells show almost no change in contrast, which means that the healthy cells are sticking strongly to the substrate, while the metastasis cells are not. It's almost floating from the substrate. So by this technique, you can visualize where the adhesion is occurring and how big the adhesion is. So this is how to visualize it. And the second thing is how to measure the force. For this purpose, we utilize the Shockwave. So this image is the bullet traveling in the air with the speed beyond the speed of sound. So when the object is traveling with that high speed, then the air will be compressed, and this will make the shock wave. Shock wave is nothing but the wave of the pressure. So if you are standing at the front of the wave edge, then you feel the pressure. So we generate this shock wave in the lab to measure the adhesion force. How? We use the laser. Especially, we use short pulse laser. When the laser is shot in the cell culture flask or cell culture dishes and away from cells, then this pulse laser can make the bubble. It's called cavitation bubble. And when this cavitation bubble is collapsed, this produces the shock wave. And it has a pressure of 1 to 10 megapascal, which is corresponding to 10 kilogram of weight onto 1 square centimeters. So it's very, very strong pressure. What happens if you hit the cells with this shock wave? If the cell is attaching to the substrate strongly, then they will stay even after it's shot by the shock wave. But if unfortunately the cell is only weakly adhered, then they will be kicked away from the surface. So by this technique, you can judge how was the adhesion strength of cells. And now I'm going to show the result of the experiment. So this is the healthy cells sitting on the artificial cell membrane. And we gave the shock wave with 10 megapascal. What happened was they stayed at the original position. So they were quite strongly attached to the substrate. We did the same experiment with the metastatic cells. And only 1 megapascal of the shock wave was strong enough to detach cells. So they are moved afterwards. So by this technique, we can distinguish and we can evaluate how strong the adhesion of cell was. So as I told you today, we are trying to evaluate the adhesion of cells, and it's visible and measurable. And as I shown, the results from the healthy cell and the metastatic cell was quite different. Therefore, we now expect that it's possible or looking at the adhesion may help the diagnosis to judge the cancerous degree and other diseases. So as I told you before, the pattern recognition is the main way to diagnose the cancer. But sometimes it's difficult because there is no quantitative parameters to characterize these kind of patterns. It's very complicated. So it's based on the experiences of the medical doctors, but sometimes they face very difficult examples. But as I shown today, the adhesion can be visible and measurable. And the advantage of these techniques was that it's possible to characterize the adhesion with the number. So those techniques were quantitative. So we expect that this will improve the accuracy of the diagnosis by looking at the adhesion of single cells. And also, we expect that this kind of approach will help us to understand why the tissue destruction has to happen in the cancer and other diseases. This understanding may help us to understand the general rule, what's happening when you get ill in the tissue. So I hope I could convince you that the adhesion is quite interesting and doing the important role in your body to keep your health. So this is all. Thank you very much.